Should we start, sir?
Yes, yes.
So, good evening to all the respected eminent panelists joined us today and all the viewers that will be joining. We are welcome to the Pharma Lok presents Pharma ki PATHSHALA, under which a thought-provoking session is entitled Animal House in Pharmacy Curriculum, Feasibility of Virtual Labs, Simulation Software, and Other Alternatives. So, under the esteemed guidance of Shri Amit Jasar, uh, National Coordinator Pharma Lok, and Dr. Santosh Verma sir, Vice President, Executive Coordinator Pharma Lok. The society continues to play a pivotal role in fostering the innovation in pharmaceutical sciences. So we believe in connecting by heart, and today's session brings some of the finest minds in pharmaceutical sciences uh, and regulatory aspects joined today, and we'll be touching the core topic of pharmacology, education, ethics, innovations, and feasibility about the subject. So, Pharmalok is distinguished for its engaging webinars and further enhancing its role in contributing in pharmaceutical science. And I'm very confident that today's session will uphold its reputation in the tradition and fostering the knowledge for the profession. So, with this, we have joined with our eminent personalities of pharmacy. So, let me introduce the eminent panelist. We have Dr. Randir Daya sir, Head of Department Pharmacology, Central University of Punjab, which will be joining us shortly. We have Professor Geer Amisak sir, Professor, Department of Pharmaceutical Sciences, University of Kashmir. We have Professor Satish Sharma sir, Vice Chancellor, Suraj Mal University, Uttarakhand. We have Tariq Mahmood sir, Professor, Faculty of Pharmacy, Integral University, Lucknow. We have Dr. Rajneesh Srivastav sir, Associate Professor, Chitkara School of Pharmacy, Punjab. We have Dr. Abdullah Sherkar sir, Associate Professor, SVKM and MIMS at Global University, Maharashtra. So I would like to welcome Santosh sir to do the introduction and to set the tone of today's discussion. Over to you, sir.
थैंक यू जुनैद जी। आज के फार्मा लोग की पाठशाला की सीरीज में हमने एनिमल हाउस इन फार्मेसी करिकुलम पोस्टरिंग ये फैसिलिटी ऑफ वर्चुअल लैब्स सिमुलेशन सॉफ्टवेयर एंड अदर अल्टरनेटिव्स वाले टॉपिक पे बात करना तय किया, जिसमें कि हमारे सभी एमिनेंट पैनलिस्ट आ चुके हैं। आपका फार्मा लोग की तरफ से बहुत सारा स्वागत। आप लोग का सुझाव हमारे लिए बहुत ज़्यादा मायने रखता है। तो आज इस पाठशाला के कड़ी में हम लोग इस बात पे ध्यान देंगे कि एनिमल हाउस और या एनिमल एक्सपेरिमेंटेशन का जो रोल हमारे फार्मेसी करिकुलम में है, खास रिसर्च वाले पार्ट पे, उसका महत्व के बारे में चर्चा की जाए और साथ ही जो वर्तमान परिदृश्य में देखा जाए कि एनिमल एक्सपेरिमेंटेशन को लेके रेगुलेटरी रिस्ट्रिक्शंस भी अच्छी खासी है। बावजूद इसके फार्माकोलॉजी का कोई भी इन्वेंशन जब तक कि हम लिविंग ऑर्गेनिज्म पे उसकी स्क्रीनिंग नहीं कर लेते, तब तक वो पूरा नहीं माना जाता। तो इन सारी चीज़ों को कि एनिमल का यूज़ करने का जो कंडीशन है, उसके अल्टरनेटिव्स हो सकते हैं, जिसके बारे में आज की कड़ी में हम अपने सभी पैनलिस्ट से के सुझाव और उनके विचार जानने की बात करेंगे। तो इसी क्रम में एक छोटी सी प्रस्तुति पीपीटी के माध्यम से आप लोग के बीच में मैं रखते हुए इसके कार्यक्रम के उद्देश्य का और इसके दिशा के बारे में आप लोग को बताना चाहूँगा। तो स्लाइड को फुल स्क्रीन पे किया जाए। आशीष जी, थैंक यू। तो आज जैसा कि आज की टॉपिक आप देख पा रहे हैं कि एनिमल हाउस इन फार्मेसी करिकुलम, feasibility of virtual lab, simulation software and other alternatives, जो कि आज फार्मेसी की पाठशाला का टॉपिक है। इसमें हम देखेंगे कि CCACA की गाइडलाइंस रिगार्डिंग ये एक्सपेरिमेंटेशन ऑफ एनिमल को लेते हुए जो चला आ रहा है, तो डेफिनेटली यहाँ पे ह्यूमैनिटीज़ को हमें टॉप प्रायोरिटी देनी है ताकि क्रुएलिटी ओवर द एनिमल किसी भी तरीके से एक्सेप्टेबल ना हो, लेकिन इसकी ज़रूरत भी है, एथिकली हम इसको आगे लेके चलें। इसमें Committee for Control and Supervision of Experiment on Animal was established under the section of Prevention of Cruelty to Animals Act 1960 and the breeding of and experiments on animal control and supervision rules of 1998, 2001 and 2006 ये रेगुलेटरी एस्पेक्ट इसके साथ जुड़ा हुआ है। इसकी ऑब्जेक्टिव जो है टू प्रमोट ह्यूमन एंड इथिकल केयर ऑफ एनिमल यूज इन बायोमेडिकल रिसर्च और साथ ही टू प्रिवेंट इन्फ्लिक्शन ऑफ अननेसेसरी पेन एंड और सफरिंग ऑन एनिमल्स, ताकि ड्यूरिंग द एक्सपेरिमेंट एंड आफ्टर द एक्सपेरिमेंट जो एनिमल है वो किसी भी तरीके के पेन को जो कि अनडिजायरेबल हो उससे हम इसको प्रिवेंट कर पाएँ। जिसमें कि जो प्रिंसिपल्स लेके चला गया उसमें एडवांसमेंट बाय न्यू डिस्कवरी है, सेविंग और प्रोमो ये प्रोलॉन्गिंग ह्यूमन लाइफ को ध्यान में रखा गया है, फिर एलिवेटिंग सफरिंग, सिग्निफिकेंट गेन ऑफ वेल बीइंग एंड लास्ट वन कम्बेटिंग एनी डिजीज ऑफ ह्यूमन एनिमल एंड प्लांट। इसको हम कहते हैं इस स्टडी एनिमल स्टडी के माध्यम से लेके चलते हैं। साथ ही प्रिंसिपल सेकंड जो है एनिमल्स लोएस्ट ऑन फिजियोलो
Phytogenetic scales.
फाइलोजेनेटिक, हाँ फाइलोजेनेटिक स्केल पे देखा जाए, जो कि ह्यूमन के जेनेटिक मेकअप से मिलता जुलता हो, ताकि जो रिसेप्टर और ड्रग के इंटरेक्शंस हैं उसकी उसका ये एक्चुअल इंटरप्रिटेशन किया जा सके। इसके अलावा मिनिमम नंबर ऑफ एनिमल का इस्तेमाल जो कि स्टैटिस्टिकल एनालिसिस को भी सक्सेसफुल बनाए, बहुत ज़्यादा नंबर ऑफ एनिमल इन अ सिंगल ग्रुप ना लें, इतना भी कम ना लें कि स्टडी आपका स्पॉयल हो जाए। तो डेफिनेटली इसको लेते हुए भी क्रिटिकल केयर करने की ज़रूरत है। इसके अलावा वैलिड रिजल्ट 95% लेवल ऑफ कॉन्फिडेंस को जो स्टैट का अपना मोटो है वो सस्टिफाई करे। इसके अलावा कंसिडरेशन ऑफ अल्टरनेटिव्स को भी देखें कि बिफोर कमिंग ऑन द एनिमल स्टडी हम वो सारे के सारे स्टेप्स को पूरा करें कि लास्ट स्टेज ही हमारा एनिमल स्टडी रहे, रेदर देन कि हमने स्टार्ट ही एनिमल स्टडी से करना है। इसमें हमारा कंप्यूटराइज्ड ड्रग डिजाइनिंग, साइमुलेशंस सॉफ्टवेयर और इसके अलावा आपका जो टिश्यू कल्चर, सेल लाइंस की बात आ रही है, वो सब सारी चीज़ें पूरी करने के बाद अल्टीमेटली लास्ट फेज में फार्माकोकाइनेटिक और फार्माकोडायनामिक्स को एस्टेब्लिश करने के लिए हमने एनिमल तक लेके जाना है। ये वाला जो प्रिंसिपल सेकंड का जो सेकंड लास्ट पॉइंट है इस बात को बताता है। इसके साउंड साउंड जस्टिफिकेशन इफ अल्टरनेटिव्स आर नॉट यूज, तो इसका भी ख्याल रखा जाए कि यदि किसी भी तरह के अल्टरनेटिव मेथडोलॉजी को हम इस्तेमाल करके हम नहीं पहुंच सकते, तब सबसे आखिरी उपाय एनिमल स्टडी का होना चाहिए। साथ ही प्रिंसिपल थर्ड है मिनिमाइजेशन ऑफ पेन और सफरिंग अनलेस ए कंट्रोलरी इज सिग्निफिकेंटली एक्सप्लेन, और साथ ही स्लाइट पेन और सफरिंग परफॉर्म्ड विद सेडेशन, एनाल्जेसिया और एनेस्थेसिया। यदि पेन की जरूरत पड़ ही रही है तो ये पेन फील ना करे तो यहाँ पे हम एनेस्थेटिक्स का इस्तेमाल करें, एनाल्जेसिया का इस्तेमाल करें, और ज़्यादा पेन हो सकता है तो एनेस्थेसिया चाहे लोकल हो या जनरल ये एनेस्थेसिया उसका इस्तेमाल करते हुए हम काम करें, ताकि एक ह्यूमनिटीज एनिमल के केयर और एक्सपेरिमेंटेशन के दौरान भी दिखे। और प्रिंसिपल फोर जो पार्ट है ये इन्वेस्टिगेटर्स आर रिस्पॉन्सिबल फॉर रिहैबिलिटेशन, यानी कि पोस्ट एक्सपेरिमेंटेशन एनिमल को हम यूं ही अनकेयर्ड ना छोड़ें, उनका रिहैबिलिटेशन पर पूरा ध्यान दें। ये भी हमारी तरह लिविंग है, और भले ही ये हमसे कुछ कह नहीं सकते जबान में, लेकिन बीइंग ए ह्यूमन आफ्टर द एक्सपेरिमेंटेशन हम इनके साथ इंसानियत की तरफ पेश आएँ, रिहैबिलिटेशन को हम पूरी साइकिल तक, ताकि इनकी जो बची हुई ज़िंदगी है वो बेहतर तरीके से गुजार पाएँ। इसके अलावा यूथनेसिया यूसिसाइड इन फॉलोइंग कंडीशन, जैसे कि एनिमल इज पैरालाइज्ड और कैन नो लॉन्गर परसीव द इन्वायरमेंट। यदि इस तरह का हुआ कि आफ्टर एक्सपेरिमेंट यदि वो पैरालाइज हो जा रहा है या किसी भी तरीके से काम लायक नहीं है, तो उसे यूथेनाइज करके उसका मर्सी किलिंग जिसे कहते हैं उस तक लेके जाएं। सेकेंड वन है एनिमल लेफ्ट विद सीवियर रिकरिंग पेन। कई मॉडल्स ऐसे होते हैं कि वो रिकवरी की तरफ नहीं जा सकते हैं, तो ऐसे कंडीशन में भी हम यूथेनेसिया को फॉलो करते हैं। इसके अलावा नॉन ये टर्मिनेशन ऑफ एनिमल वुंड बी लाइफ थ्रेटनिंग टू ह्यूमन। कई बार हम माइक्रो ऑर्गेनिज्म के थ्रू या ऐसे सेल कल्चर को इंड्यूस करके मॉडल डेवलप करते हैं जो कि यदि सरवाइव करता रहे और उसका ख्याल ना रखा जा पाए तो ये ह्यूमन रेसेस में भी पैथोजेनिसिटी को डेवलप कर सकता है। ऐसे सारे केसेस में हम यूथेनेसिया को फॉलो करते हैं, मर्सी किलिंग को फॉलो करते हैं एनिमल स्टडी के दौरान। तो इस तरीके से मैं ये चारों प्रिंसिपल को आप लोगों के सामने रखा। अब एमिनेंट पैनलिस्ट से गुजारिश है कि जुनैद जी आप इसको कंटिन्यू करें।
Thank you so much for setting the tone for the discussion, sir. Once again, thank you. And we will start with, as one of the panelists have not joined yet. So we will start with Professor Geer, sir, for the first question, sir. Can simulation platforms provide reliable experimental results that the student can interpret and apply as they would with real life animal models?
Thank you very much, Mr. Junaid Tantri ji, Dr. Santosh Verma ji, all other esteemed panelists in this panel discussion on in this Samvad of Pharma Lok today. A very good evening to all of you. The simulated animal experiment platforms do provide reliable, pedagogically sound, and ethically viable alternatives to real animal models. But the educational context becomes very important. In what context we are talking about, particularly for pharmacology and physiology, because their utility is context specific, and it is important to distinguish between educational reliability of these simulated models for teaching and learning and their scientific validity for research and regulatory drug testing. So far as the teaching and learning is concerned, the demonstration for undergraduate students, their reliability is good. They are reliable, pedagogically sound, and ethically viable alternatives. As just now mentioned by Santosh sir, that these are important things which have been propagated through various enactments, laws, that we have to resort to alternatives. And it has also been proven through scientific studies that they are reliable and they are reproducible, their precision is good. But no, the studies done by Patil et al. In 2013 says that 78% of students found these simulated experiments more understandable than real life experiments. Another study done by Ravindran et al. From JIPMER says that students using XPalm showed improved conceptual clarity and retention. Another study by Badiol et al. From Ames in 2009 says no significant difference in exam performance was seen between students trained with computer-aided learning and those with live animals. While as another study in 2017 by Gupta et al. Says that computer-aided learning users reported less stress, higher satisfaction, and deeper understanding of pharmacokinetics. So for teaching learning process, for routine demonstration of UG students, they are reliable, but their scientific validity is questionable, that we cannot use animal models for research purpose because of their biological variability. In research, we use animals of different sex, different age groups, or stress effects on animals. We have to observe the toxic effects. That is not possible on simulated these experiments as on today. So the adverse effects become unpredictable. And then the pharmacokinetics, the absorption, metabolism, excretion in full depth does not become possible with these simulated animal experiments. Therefore, there is this issue of interpretation caveat also, and these are not valid for drug approval. If we do research on simulated experiments, they will not be considered for clinical trial, preclinical trials, and they will not be considered for drug approvals. So, simulated platforms are not a significant or reliable replacement for in vivo testing when it comes to drug discovery, research, toxicology research, clinical pharmacology, pharmacokinetic research, because no, they do not produce any novel data. These simulated software do not produce any new data, novel data, and therefore cannot be used for regulatory submission. They Most of the times it so happens that if one group of students uses the animal simulated experiments, they get the similar results as that of another group, and very fast they are able to complete these experiments. So there is a lot of scope for improvement of the simulated software which are available as of today. And we need to work a lot to make them more robust, more effective, more variable, more student friendly. And we need to work a lot to increase their specificity, sensitivity, reliability, reproducibility, scalability, efficacy, and accuracy. These are the things which are of paramount importance in experimentation. Whether an experiment is specific, sensitive, reliable, reproducible, whether it can be scaled to another level, whether it is efficacious and accurate. So precision is important, accuracy is important, which is somewhat lacking in simulated animal experiments. So we need to know when we need we even today when we see that when our students observe the videos on these simulated experiments, the videos are not of good quality, they are not elaborate, so the students do not get to see the full dissection process, they do not get to see the full videos. So there is a lot of scope, although they are viable, they are reliable for these teaching learning at UG level demonstration purpose, but they need to be improved drastically. So we need to enhance their robustness and scientific accuracy by incorporating pharmacokinetic and pharmacodynamic models, PKPD models, where we can study ADME, absorption, distribution, metabolism, and excretion of drugs. We need to integrate these simulated models with real world PKPD modeling using compartment models. Just like PK, there is another software like PK Sim or CYP. They have integrated real world PKPD models with compartment models so that the students can be allowed to simulate drug interactions. They can be allowed to undergo these BABE studies, half life studies, and they can better visualize variability in drug responses on the basis of age, sex, and other variables. And we also need to add genetic variability, the variability in stochastic simulations that randomize responses slightly to different biological uncertainty. So genetically modified models need to be incorporated. So omics related modeling, informatics related modeling, simulation based computational modeling. There is a lot of scope on improvement of these simulated animal experiments. Today, when we give these experiments on simulated software to our students, they finish it within 10 or 15 minutes. So every time they get the similar results and not much difference is seen. So with the passage of time, they lose interest. And then for the examination purpose also, they're not very robust as of today. For examination purpose, they don't put in a lot of variability, a lot of challenge for the students. And even the different models need to be given for each experiment. On the sidelines of the experiment, we need to give different models so that the students are fully able to understand how exactly it works, because the main aim and purpose of these simulated experiments is to understand the mechanism of action of drugs. So we need to give different models apart from the one which has been demonstrated in the particular software. Then data generation, statistical analysis, and interpretation also should be incorporated into the simulated experiments. The care based scenarios and the other statistical analysis also should be incorporated. We need to make these platforms more student friendly by using gamification approach. See, in gamification approach, when the student crosses different levels, they get different rewards and they open up different these pathways and algorithms. So that makes it quite interesting and fun based for the students. Nowadays, the NEP also advocates gamification based more perspective or model of teaching learning. And we need to layer complexity for different academic levels, and for we also need to make it multilingual, so that there are already many software which are multilingual in nature, so that people, students from southern part of India can visualize it in their own indigenous languages, and northern part of India can visualize in their own local languages. So that way we can improve the specificity, sensitivity, reliability, and reproducibility of the experiments, and increase their consistency and randomization. We can also make them cloud based so that they are more accessible. Cloud based platforms need to be used, and so that they become plug-in ready for new modular learning. and validation and feedback loops also need to be incorporated, so that periodic user surveys and learning outcome mapping can also become possible. So we need to take help of the computer professionals, software developers, to make them more robust, more modern, more contemporary, and more effective, more reliable. And so that PKPD modeling, drug interactions, and the variability in genetics, variability in sex and age, and then modular learning and real experimental data sets become possible. So these kind of changes need to be made even for demonstration purpose. But so far as research is concerned, for research, definitely we need to make use of animals, because clinical trials, that data has to be generated, new data has to be generated, new data cannot be generated on simulated software. But then, of course, there are these culture cell culture models, and there are in vitro models which also need to be incorporated. Of course, I'm all for it to minimize the use of animals wherever possible, to first start with in vitro experiments, then move on to the preclinical studies. That will make the preclinical studies much better and faster. And we need to incorporate these in vitro models, cell culture based models, stem cell based techniques will make our students more efficient, and they will be in a process to learn better, because these are the new agents. Everything in future will be done on simulated basis. See, nowadays we have huge infrastructure in our educational institutions, but it is believed that this infrastructure will not be needed, because in future more and more AI based techniques, more and more AI tutors will be used by the students, and the simulated atmosphere will be used. That is the digital twins, for example, the technology of digital twins. Nowadays, now at present, the clinical trials take 14 to 20 years. They consume billions of dollars, but these same clinical trials can be done within a span of few months on the basis of digital twins. When we use informatics based approaches, or omics based approaches, biosignature of the humans will be available to us. So we can do clinical trials also on simulated software, which will be possible through digital twins. So these new concepts need to be taught to the students. They need to be incorporated into the curriculum, and we need to adopt these techniques as much as possible. But at the same time, the importance of real life use of animals in preclinical testing and preclinical studies cannot be undermined. That should be continued. For demonstration purpose, of course, we can reduce the use of animals, which are very reliable, effective, and safe to use. Thank you very much. If possible, I will give more comments later.
Thank you so much for giving so clarity in the answer, sir. Thank you for speaking that yes, these softwares are reliable for UG programs, but for PG, definitely we need more integrated and more robust softwares. And you also have given us that various references, Patel 2013, Patel ETL, Ravindran ETL, which has proved that. These have been the students have given a positive response regarding the clarity of concepts with these apps. Thank you for giving us deeper understanding, sir, and thank you for talking
Thank
about
you.
PKPD models also, that omics needs to be upgraded, and the most important thing which you spoke is the gamification of softwares. I think it's an important point there, so that it can be relevant to the students also. Thank you, sir. So we'll move to the next question. Uh, the next question will be to Professor Satish Sharma, sir. Sir, the question is with increasing concerns about animal welfare and environmental stressors, noise affecting lab animals, how ethical and sustainable it is to continue using live animals in routine pharmacy training?
Okay. Thank you, Mr. Junaid. And good evening to all respected delegates present here. It's a good question out. And so I will start my talk from here about training of pharmacy students on living animals. So training, I will discuss. Training is an important aspect for getting success in anything. I will say training is a gateway to research, I will say. And training, if we conduct training, then the individual is trained with the skills. And skills are in national education policy 20.
Sorry to interrupt you, sir. Sorry to interrupt.
Hello?
I think there's a audio glitch, sir.
I'm not audible.
सर आपकी आवाज
Yes, sir.
ब्रेक हो रही है।
Break हो रही है
हाँ,
sir.
आपकी आवाज इंटरप्ट हो रही थी। एक बार जरा। हाँ
Am I audible? Am I
हाँ,
audible
यू आर
now?
ऑडिबल।
Again, there is an interrupt, sir. Again, there is a little interruption. You can rejoin, sir. You can. I think you can rejoin once again.
Hello.
सर एक बार
नेटवर्क
ये
इशू
लॉग
है
आउट
सर,
होके
नेटवर्क
दोबारा।
इशू।
हाँ,
Network issue
लॉग
होगा
इन
sir. It's a
करें।
network issue. This can
हम्म
happen.
आपका वीडियो तो क्लियर आ रहा है,
Uh,
आवाज ग्लिचिंग कर रही है।
आवाज नहीं आ रहा? अब आ रहा है आवाज?
हाँ, आवाज तो आ रही है सर।
आ रहा है?
हाँ
अच्छा
सर,
अच्छा.
हाँ हाँ।
So what I would like to say
Much.
that training is an important aspect to achieve success in any domain, whether it's whether it's academic field, whether it's research field. So I have been questioned about training. So training is an important aspect. Training on living animals. So training on living animals is also important. Of course, it's a challenging task, especially as has been mentioned by Professor Gee, that we are living in AI era. It's a new era. Everything is being compared with AI, and everybody is crazy about, excited about going for AI alternatives. But I will reckon that we cannot learn swimming by watching the videos. If we only keep on watching videos, we cannot become a swimmer. To become a swimmer, we need to dive into the swimming pool. Similarly, we cannot become Sachin Tendulkar merely by watching the videos of Sachin Tendulkar. So what I'm trying to emphasize is that previously also when we are students and up till now who are studying, animal house was always given, a column was always given when we used to fill this manual format, SIF we call it, standard inspection form for PCI. So animal house was always a necessity. But now we have received an order from PCI in 2019 that animal house is not mandatory in cases when it is feasible to conduct experiments through simulated process, simulated software. That is right. But this animal experimentation is an important aspect of drug discovery as well. We cannot deny this. And all these drugs, most of the drugs which are discovered, invented, have come through preclinical studies, that is through animal experimentation. If we are formulating something, then to evaluate this preparation, we need to go for pharmacological evaluation through preclinical studies. So animals should be given top priority. Animals, we should revive back the culture of doing experiments on animals. Because by simulations, as has been mentioned by Professor Geer also, that simulations are okay up till demonstration purpose. But when we are doing research in farm level, then obviously one cannot enter directly into research. As I have already mentioned, that we need to train ourselves before going for actual research on any field. So I strongly reckon, I strongly support that animal house should be given due importance. Experimentation on animals should be given due importance, and especially in the N-farm level. And I appeal to PCI and I suggest that PCI should reconsider the aspect of considering animal house as an important part of pharmacy curriculum. But previously also, it is as good as laboratories. It is one of the laboratories for doing experiments. It is one of the laboratories. It is as good as laboratory of pharmaceutical chemistry or pharmaceutical analysis. In pharmaceutical analysis, at B-Pharm level, we get trained about use of spectrophotometers, NMR, and all these things. At M-Pharm level, we start working, we start doing research. So first of all, we need to get ourselves trained. Now comes the question of feasibility. Question of feasibility mentioned, Mr. Junaid, you mentioned in your question about the feasibility, whether it is feasible to perform experiment in this scenario. Scenario means you have mentioned first part of the question, animal welfare, considering the concern of animal welfare and considering the aspects of stressors, means environmental stressors. So what we can do is, if as has been explained by Professor Verma in his presentation, that there are ethical principles. There are 5 ethical principles suggested by CPCSEA. If we follow all these 5 principles, then it becomes viable, then it becomes feasible, and we have mentioned about whether it is sustainable or not. I will mention in NEP 2020, it is clearly mentioned that we have to provide sustainable education by providing holistic, skill-based education to everyone. This is complete. Objective is to bring to reinstate India as Jagat Guru by providing skill-based education. So skill-based education at all level, primary level, secondary level, and in all domains also. So we cannot develop skills by simulated software. We can develop skills. We can understand a little bit how the practical is being done. But once we will come in the animal house, then we will see the skills. What skills I'm talking about is, we will learn how animals behave. We will learn how animals are transported. We will learn how animals are handled, and we will also learn the full dissection procedure, how to dissect it, how to take out the tissue, and we will also learn the location of a particular tissue and organ. We will come real life situation. Similarly, we will learn the sorting of the tissue. We will learn different staging methods as well. We will learn practically. We will learn the skills. If we jump directly into doing higher level research without knowing the basics of the animals, then the reproducibility, precision, accuracy is always in doubt, because the candidate is directly entering into research. In pharmaceutical chemistry also, in pharmaceutical analysis also, there are sophisticated instruments. Unless you learn these instruments at B.Pharm level, you cannot do the actual research work on IR, HPLC, mass spectra, or whatever. So all these sophisticated instruments are meant to be learned at B.Pharm level, or at M.Pharm first year level. So I reckon that this animal house facility should be mandatory, and secondly, this M.Pharm facility should be mandatory at M.Pharm level. Should not be mandatory at B.Pharm level. Very well explained by Professor Geer that we should minimize the use of animals. That is important aspect. So once we will follow these ethical principles, which you Professor Verma, you explained very beautifully, then it becomes very feasible. So we can say that it is quite possible. And sustainability aspect. What is sustainability? Sustainability means, if we have skills, it means it is sustainable education, because skills will be useful for whole life. Once we learn animal experimentation through training, then this training will be an asset. It will act as an asset for that particular researcher, and this asset will be useful for gaining experience, for succeeding in all fields, whether it is R&D field or any field. He can go in contract research organizations also. Once he know, so this is an important skill. Animal experimentation, use of animals is an important skill, because students will obviously consider himself that I have learned something great in pharmacology. Not only in pharmacology, I will as per NEP 2020 also is mentioned that education should be holistic. Education should be interdisciplinary. So when a particular person, there are 10, 11 branches in pharmacology. Suppose there is pharmaceutical chemistry branch. So for doing advanced research in pharmaceutical chemistry, the researcher will first synthesize the derivatives, then he or she should evaluate its activity. He should be able to evaluate for what purpose he synthesized. Suppose some derivative or compound is synthesized for anti-inflammatory activity, then he can go further for advanced studies, means advanced research. There will be interdisciplinary research. This is called as interdisciplinary research. So he can conduct research simultaneously in pharmaceutical chemistry as well as in pharmacology and at MPharm level. Being a student of pharmaceutical chemistry, he will learn the skills of pharmacology as well. This is very important in the current scenario where everyone is talking about interdisciplinary research, multidisciplinary education. So in this way, I will mention again in pharmaceutics also, when somebody is doing research, M.Pharm in pharmaceutics, then the research candidate will develop a formulation. Whether this formulation is effective or not, the purpose of this formulation is to increase bioavailability, whatever the purpose is there, to study the mechanism of action, to increase the efficacy, to increase the potency. So for that purpose, obviously preclinical studies are required. So pharmaceutics, M.Pharm pharmaceutics researcher can go further for advanced research at M.Pharm level itself. So at M.Pharm level, he will be exposed. He will be following the principles of national education policy. National education policy always considers, always recommends that we should go for interdisciplinary education. We should go for multidisciplinary education. We should go for interdisciplinary research. So at M.Pharm level, if animal house is available with that institute, then it becomes an asset. Anybody, any candidate, any researcher, irrespective of the field from which he or she is doing M.Pharm, he will have something extra, something research lab, that I will call animal house research lab. It is an important research lab. It is an important skill generation lab. So animals are very important, but at the same time, we should follow. If we follow the ethical principles, means minimize the use of animals, means we should take all the necessary steps to minimize the pain, sufferings, distress, and we should give all the natural facilities. If we give all the natural environment to the animals at animal house, then obviously we can meet the guidelines. We are following the guidelines. Not only guidelines, but it is our human. It is our duty. It's our moral duty to treat every creature as equal. We should not we should not make any creature of the world to suffer. So this is our moral duty as well, as irrespective of guidelines also. And then you also mentioned in your question the aspect of environmental stressors, like PCCSA has given guidelines for establishing animal house. So we should meticulously, honestly, ethically, morally, should follow all these guidelines. We should follow, and we should impress upon in the minds of the management that if we will establish a good animal house as per the guidelines, then obviously we can invite other institutes also. We can get other institutes proposals also for research. So it will help in revenue generations as well. So revenue generation aspect is one. Other is enhancement of research. Second aspect. So physical aspect is there. One part is there, which the last part of my talk is, which you mentioned in your question about environmental stresses. For example, noise pollution, noise. How to deal? So when noise is there, obviously the animals are going to suffer from physiological ailments. their physiological system will be deranged, no doubt. They will suffer. Their CNS may be disturbed, their neuroendocrine system may be deranged. So it's very important. Noise affects not only auditory system, but non-auditory effects are also being noticed. It affects our reproductive cycle as well. It noise affects our circadian rhythm as well. Noise as well as light both. So again, I will say that as pharmacological experts, we should take these things sincerely. Sincerely means seriously. Read the guidelines, and we should try to convince our management. Actual guidelines we should follow. What I'm trying to impress upon is here that first of all, so I will talk very briefly about noise. How to deal with noise stress. Obviously, it is an important aspect. It is adversely affecting the health of the animals. So when we are deciding, we are selecting the site of animal house, then we should think about the thing that this animal house should be away from the main road, main highways, from main movement places. It should be, I mean to say that animal house should be at a separate place, at an isolated place. Isolated place doesn't mean that it should be an unsafe place. Isolated place and a safe place. Okay, but movement should be limited at that place. We should meticulously select a site at our campus building. So so if we can select a separate isolated site, then obviously noise of movements, noise of roads, traffic will be minimized. So there are two types of noise which are disturbing the animal. One noise is outside the animal house. So we can, by selecting a suitable place, isolated place of animal house, we can minimize the noise level. Second part, noise in animal house. Obviously, there are so many types of sounds, and you will also agree with the fact that animals are very sensitive to hearing. We cannot listen the sound, but animals can listen the sound. So we should install the sound detectors, sound meters, sound controllers in our animal house. How to minimize
Absolutely.
the sound or noise in animal house? For that, we need to train all the staff members. We need to train all the researchers there. Training for sensible doing, sensible talking, sensible processing. For example, in an animal house, when any staff enters inside an animal house, then the doors of the animal house should be opened gently, not forcibly. So if we'll open the door gently, then you can minimize the sound. Similarly, when you are leaving the animal house, you will close the door. You close the door sensibly, gently. So training. Then secondly, when you are wearing the boots, boots should be soft. The sole of the boot should be soft, maybe rubber sole. So when we are working, sometimes in the current scenario, we have seen so many fashion-made boots are wearing, and you can hear so much loud sound. So this is again disturbing to the animal. So we have to keep in mind all these aspects. Noise control
Satish
is
sir,
similarly. So
sorry to
hello,
interrupt, sir. Sorry to
yes
interrupt,
sir.
sir. We can get back if you concise because there are one round we want to complete. Then we can discuss once again for the second round, sir.
Okay, okay. So lastly, what I meant to say is, in summary or within a few seconds, I will say that it is very feasible to conduct animal experiments on live animals at postgraduate level, and animal house should be made mandatory for those institutions which are going to start M-Farm in any discipline. This is my aspect. Thank you so much.
Thank you so much, sir. Thank you so much. There will be a separate lecture for this with you, sir. And Randeep sir has also joined. Welcome, sir. So
Thank
thank
you.
you for speaking, sir, on importance of training, Professor Satish sir. Thank you for giving us the idea that simulations can be used, but it needs a revival. It needs certain any institution who is having animal house should follow the basic principles so that the animals also should not be harmed. Thank you, sir. My next question will be to continuing with uh, the discussion. So we have one more question for Dr. Tariq Mahmood, sir. Sir, what are the regulatory challenges and infrastructural gaps in implementing simulation-based alternatives in Indian pharmacy college, especially in source resource limited college?
Thank you, Junaid. Uh, Good evening to all respected dignitaries present here. Uh, the question for me that is having two parts basically. One is, as Junaid asked me, that regulatory challenges, and the second part of that question is the infrastructural gap basically regarding the simulation-based alternative in Indian pharmacy colleges. So, what I want to say here actually that is two-edged process basically. The first thing is the regulatory changes, which is very important, and because I think that 2 points are basically the two main hurdles to apply that simulation based, or you can say the computer aided learning based education, especially in the pharmacology part, which is the main concern for today's talk. As Professor Gir sir very rightly said, that the present time and the future is the era of that simulation based, and I totally agree with that. So we can, and as recently Professor Satish Sharma said that animal house is necessary and animal experimentation should be must. Yes, of course, but the main important part is regarding the teaching and learning, as Professor Gill said. The research part, of course, everybody knows that for research purpose, we need the practical hands-on training. Either it is related to the pharmacology or any discipline, we need to hands-on training, not only the simulated part concern. But for academic purpose, or you can say teaching learning purpose, we start from the graduation, as NEP also mentions that which should be available for any discipline. So the simulation based education is very hot topic presently to discuss regarding especially in the pharmacy. So I come back to the question. The regulatory changes is, when we discuss about that, the first point is the lack of mandate integration. What it means basically, it is related to the, you can say, the policy, or you can say the a particular guideline from the council or previously the ICT, that do not provide any explicit mandate or framework for incorporating simulation into pharmacy curricula. This lack of regulatory clarity has led most of the institute to view simulation as optional rather than essential. Some year back, a very common software, we all know that XFARM, there is a communication from PCI that when the animal house experimentation has been banned in prohibited for the UG program, we should move to that pharmacology practicals on the simulated one, a very basic software of that one. But except from this, there is no clear mandate or guideline regarding the whole simulation lab or simulation software. Here we all agree on that one. Simulation doesn't mean that only the simulation software. It is a whole science. When we talk about simulation, it start with very basic software to you can say the 3D structures of the animal, and the 3D labs of the models of the virtual screening. As my colleague Daya sir also joined, he also a very prominent person regarding the pharmacology. He knows that one. The future of that one is the 3D animal models. So we need a particular guideline and mandate regarding that one. The second one is the rigid curriculum and assessment models. That is the second problem basically regarding when we talk about that regulations. Basically, our UG, when we discuss our other UG program or PG program, basically, I'm sorry to say that, but it is basically the theoretical oriented program. Of course, there are practicals, but when we observe, you go to the practical syllabus of any pharmacology year, either in the third year or in the final year or in the UG. They are not aligned with the first thing, they are not aligned with the theoretical part of that subject, and the second thing is that one, there are no standard assessment protocols for simulation based learning in there, and there are no, you can say, rubric system for the evaluation of that assessment model of the simulation experiment. So that this is the second point, and the third point in this is that absence of accreditation incentive. That is very important one. A very common example, we take the NBA or we go for NAC. So there is no such specific, you can say, points, reward point or any other thing which gives the simulation based teaching infrastructure living institution with living little institution motivation to invest. That is a very important one. The same with that council, and the same with that accreditation body. If you have the simulated one, they say it's okay, it's good. Okay, yes, they make the yes or no option. They make that, but there is no such one that if any college have that certain more sophisticated software, as mentioned by many software mentioned by the Gir sir, expect from the very basic software. So they give the advantage or extra point on that one. There are number of software available, but the thing is that they are not mandatory as part of curriculum, or they are not mandatory as part of their assessment or accreditation. So the third, or then not the third, that is the fourth. The fourth point is that the limited faculty trainings. That one is very, I think not very, it is the most important part. Introducing a simulated software or simulated lab in any institution is not the thing that the work has been completed. The most important part is the teacher training on that one. And I think last year I attended an IFDP in any college of the South. I think I do not remember the name. They organized the FTP on that one training of the faculty on different simulation software. That was very good training. So I think it is a very important part, and every college, I think, and I request all pharmacology teachers of UG and PG find out that this type of DPs or any other programs or any other platform, any MOOCs platform or any other platform, and do that training and hands-on on that running of that similar different types of PKPD software or pharmacology simulation software and simulation labs. There are the simulation labs out there, a complete CNS simulation lab, complete cardiovascular simulation labs, complete GIT simulation lab, complete reproductive system simulation lab, and they provide the training for faculty at least. So go for that training and learn that one. That is very important one, and I at the college. Authorities also please motivate to the faculty to go for that one. It is very important one, but because before giving to that training, or you can say that simulation education to students, first we learn that one effectively. So that actually that that 4 points basically comprises related to that regulation. And now the second part of the question, which is related to infrastructure gap, that is actually the bitter and crude reality of that one, because that the initial and maintenance cost, both the initial cost and maintenance cost is very high, except that very basic software as I discussed earlier, which are the, you can say, cheap one. And every college have that, I think every college has that certain one or two the software, and they fulfill the norms, and they run that UG pharmacology on that software only. So that is not the point basically. So establishment of simulation labs requires significant capital investment, software and AV equipment For college in rural areas or semi-urban areas, these costs are often very difficult to obtain. Moreover, maintenance requires ongoing technical support, which is usually unavailable and very costly one. And that second one is the lack of digital infrastructure. When we move to the higher level of the simulated softwares or simulated labs, it needs a basic technology, a higher level of digital technology. Start with a very stable internet, modern computers, power backups, simulation platforms, AR or VR or cloud-based software, as Professor Geer also discussed regarding that cloud-based softwares. So it is very difficult for a simple UG college to maintain all that one because it involves a lot of capital. So we need to discuss regarding that one. And the third thing is that shortage of the trained personnel. It is also a very important point. If we purchase any, suppose that any higher end of software, PKPD software, so we need at least one faculty and some research scholars, some junior faculty which are on train to operate their simulated software or simulation lab. So there is it is a very clear one point. There is a nationwide deficit on simulation experts, instructional designers, and trained faculty. We need a force regarding that. See the examples of the computer application of the CS. They have the huge manpower. They are the expert, huge number of expert regarding their simulation labs. Another example is the civil engineering. There are civil engineering labs, and they have a lot of experts which are the expert in that simulation labs. So we need to point on that point also. And the fourth one is the cultural and language barrier also. Many rural institutions face resistance from faculty and students unfamiliar with the computer, unfamiliar with the language, unfamiliar with the digital tools, and it is very difficult to uh, make with these people, either with the faculty or a student, to make a common platform on that one for that higher digital level, you can say computer aided learning pedagogy techniques. So we should focus on that one. Basic preliminary, or you can say the basic training is required for both teachers and faculty, both teachers and student also. So the key points from my side regarding this question is the first thing is that policy and regulatory reforms. There should be a clear mandate from the and the second one is curricular modernization, including that simulation pedagogy include that. And third one is the recognition by accreditation agencies. Any college or any faculty, we have run that simulation software or higher end of simulation labs or simulation organ study. They should be appreciated on that one. Mandatory faculty upskilling, that is very important point. Again, I discussed that one. There are national program on STEM and MOOCs portal or any other from the NIPER also, from the JIPMER also, and many other online platform. Go for that one and upskill regarding that simulation hands-on. And practical solution is that low cost or open source simulation tools, the hybrid models. Hybrid model is also a very good alternative on that one. Use hybrid model means use mixed method of simulation combining physical model with the digital and virtual simulation lab, that is known as vSim labs, which lowers the cost of that one. And the third point regarding that solution is that PPP, that is public-private partnership, encourage collaboration between pharmacy colleges, edtech startups, and CROs, which initiate the funds to establish the labs, sophisticated labs, at least with the good universities and with a good infrastructure and with the pharmacology and pharmacy education. And targeted funding and grants, it is very important one. Allocated specific grants under the government scheme like RUSA or TechUp, that two programs which provide the grant regarding that simulation pharmacology labs. One of my friend got the fund from that Tech IP in the last year regarding that simulation labs.
and localized software development, promote the development of multilingual simulation software with regional language interface to improve accessibility and reduce resistance. So the conclusion of that question, I conclude my answer in a few lines, that is the integration of simulation based education into pharmacy Indian Pharmacy College is not just a pedagogical update. Basically, it is a necessity at present time. I totally agree with the Gear sir on that point. It is a necessity in the modern pharmaceutical training. However, achieving this requires deliberate policy shift, infrastructure investment, focus, scalable solution, and bridging the rural-urban divide on that one with the right support mechanism and collaborative effort. Simulation-based learning can become a transformable tool in shaping competent and confident pharmacy graduates across the India. Thank you so much for patient hearing.
Thank you so much, sir, for giving us the real check regarding the current status of the animal house, or whether you talk about importance of hands-on training. Of course, we agree on the point, and you also spoke about lack of mandate or regulatory. There's a lack of mandate regarding the regulatory clarity on guidelines, particularly in context to the animal house. You also talk about that simulation means whole science from basic to 3D. The good institutes should go for these 3D model labs, and particularly with the advancement of the persons, also those who will be advanced in the training also. And secondly, also highlighted the very drawback point, that is rigid curriculum, which definitely at the end of it will be the point to be discussed. And one more thing you highlighted, sir, the current scenario is not aligning with the theoretical part. The experiments which have been provided in the practical of, you particularly mentioned third sem and other B farm semesters, they are not aligning with the theoretical part. So yes, and you also one more important point: NBA and NAC doesn't give any specific credits. I think we have Geer sir, which sir will at the end will highlight on this point also, that NBA and NAC, there is no specific point rewards which which they give to the institution. Yes, sir.
Mr. Junaid, if I could make a quick comment, just small comment. I completely concur with Mr. Tariq Abdullah Saab, Tariq Mahmood Saab, what he has said. And because it has opened a larger debate of the importance of simulated software, because today we are discussing strictly in the context of animal house and in the context of pharmacology, experiments on pharmacology. But as rightly mentioned by Tariq Mahmood Saab, the importance of simulated experiments is way beyond pharmacology. And this is the comment which I would like to make at this instance, that it will have more applications even in pharmaceutical technology in future, in pharmaceutics, in pharmaceutical chemistry, wherein the manufacturing scenarios could be simulated in the laboratories, and students could actually play with it, and they could see how the different machineries work and how they have to be handled. And this is what on the other day I was attending a workshop on skill development, which was being organized by the Ministry of Skill Development, and they were endorsing the use of simulations even in apprenticeship, even in internship, and they were endorsing the use of simulations in everywhere, in manufacturing, in the practicals, in lab simulations, where the industry expert could also be present, student and the teacher could also be present, and together they can work on certain problem based learning. So because the experiential learning is gaining prominence, the project based learning is gaining prominence, so this has opened a wider question, that has the time come when we should discuss about simulations in other specializations outside of pharmacology, because so far it has been restricted or limited to only pharmacology. But now the time has come when we should also think about simulations beyond, because last year the Nobel laureate in chemistry, Demis Hassabis, he unfolded 200 million protein structures, and on that basis he said that it would have taken 10 billion years of PhD to unfold those 200 million protein structures, which he has done in just 2 years. And that's why he was given Google DeepMind, made as the CEO of Google D, and was awarded Nobel Prize in chemistry. So things are changing pretty fast, and we need to align ourselves to these changing circumstances. I think the right time has come when we need to adopt these emerging, evolving technologies of 21st century, which are AI based, AI driven, and wherein the simulations have a very important role to play. Thank you very much.
Thank you, sir.
Thank you for adding to this. Definitely, sir. Thank you. And we will come to the next question to Dr. Rajneesh, sir. We have a question that how do simulation tools prepare students for higher education or research role, and can they adequately replicate real-time decision making and procedural accuracy? Rajneesh, sir. I think Rajneesh sir will join us once again. So I will come to the next question to Dr. Abdullah Shirkar sir. Sir, could a blended approach combining limited live experiments with advanced virtual training offer a feasible and pedagogically sound alternative in pharmacy education?
First of all, thank you for inviting me as a expert for to give the insight, particularly on the blended learning and which is by the live experiment plus virtual advanced virtual training. But my answer or my insight based upon two criteria. First, as far as the UGC is concerned, undergraduate curriculum is concerned, yes, it can be used, but it has 3 to 4 justification. If you see the governing criteria or the rules and regulation given by the CCSEA or the PCI, they are working on the 3R principle. But first one is the replacement, reduction, and refining. So this case, this will reduce or the minimize the number of animals used in the experiments. Second thing, if you use the live experiment, limited, but it is a limited experiment. Say example, how to give the route, perform the route of drug administration, how to isolate out the biological samples. So, or the behavioral observation, if you want to see. In that case, this provide the efficient hands-on training to the students. Third, as far as the outcome-based education is concerned, which is given by the rules and regulation, it is mentioned in the rules and regulation of the PCI. So it creates critical thinking among the students. And fourth point is that depend upon the management or the institutions, they cannot bear the much amount on that. The use of the animals, so limited use of the animals reduces their cost. Second thing, as per the insight of the Dr. Professor Gir sir, I totally agree with that. We should go in the current scenario. How? One is the use of the microarray technique while doing preclinical experiment, or suddenly going into the preclinical research. Second thing, we have to identify the use the techniques such as network pharmacology, where we can identify exact the probable pathways based upon your drug or the phytoconstituents. So this will create minimize the errors in your experiment, and this is totally applicable for the young from postgraduate level. So yes, up to the UG level, a limited experiment should be there, particularly live experiment. But the question is that the if advanced virtual labs, it is very difficult to establish in all colleges. So this will create hurdles in the blended learning. Second thing, if we are using the as per insight given by the Professor Kid, that the video quality of the streaming, live streaming, these are not good. So student cannot understand the basic, say example, how to give the route of administration. In that, the this live demonstration will increase their hands-on training. So these are my points. Blended learning can be used, but with the conditions. Thank you, sir.
Thank you.
Audible, sir? Yes.
Yes, sir. Yes, sir. Thank you,
Okay,
sir.
this much I want to
Yes, sir.
say.
Have you completed, sir?
Yes, sir. Yes, sir.
Yeah, thank you once again. Thank you so much for giving clarity regarding this. Thank you for highlighting the point regarding 3 R's that we should focus on, and we should also focus on outcome-based education. Also, thank you for talking about that institutions are also dependent. The management is also trying to cost cut by using less animals or infrastructure. Regarding this, we will come this point at the end also, as other delegates also. So identifying, you also give the important point that we all should remember that to identify techniques like network pharmacology and softwares can help us to minimize errors. First we should go with the in vitro, then rather than going directly to preclinical. Thank you so much for the brief answer to this. We'll come to Dr. Randhir, sir. Sir, we have a question regarding uh, to what extent are virtual labs and simulation software effectively in developing practical pharmacology skills in students compared to traditional animal experiments that has been going on?
Okay, and that is a very interesting and very close question to me. And actually, I have heard all the other speakers, and as Professor Gir said, that lots of innovations are there, lots of new techniques are coming up, and virtually we can do each and everything. But being a classical pharmacologist, I will simply say that whatever techniques are there, they are serving all the purposes. Because as Professor Mahmood had said, that although we can have all these techniques, but we need a good infrastructure, we need a lot of cost is involved, as well as skilled persons. These are one of the most important factor, that if, although that you are having all the things, but you are not having a technical person or a skilled person, so it will be very difficult to get the outcome. So if I compare that what the students can get from the virtual learning as compared to the classical method in the pharmacology, the classical method of pharmacology is still, I will say, the golden one. Because whatever techniques are there, if the animals are not able to handle the animals, sorry, students are not able to handle the animals, they don't have the exposure. Because what they are seeing, because suppose in virtual experiments you have given an experiment that you can check the effect of the drug, but that is totally a calculated one. In case of pharmacology, what is there, these are the biological variants. Means that drugs are there, drugs effects are there, but it is not necessary that much drug will show that much effect, because there are very high chances of variance. And that's why when we are using the animals in case of virtual labs, what you have given the drug, you will see that. But sometimes what happens that suppose that you are given a drug, you want to check the onset of action, that as per virtual lab or as per software, in case of oral administration, you will get the response within 30 minutes or the 30 to 45 minutes. But in case of if you are using the animal, there may be variation, because in some of the animals they are, you can say that you will get some response, but in case of some animals you will get a delay of 5 to 7 minutes. Means average will be there, and this will give a real practical application of the pharmacology experiments. Because whatever is there into the, you can say, software, that is not practically possible all the time. So when you are doing with the experiments and the studies in the pharmacology, especially in case of postgraduate courses, as well as into the PhD, because these are totally based upon the animals. And we cannot simply rely upon this that software will serve all the purpose. No doubt that for the demonstration purpose, they are good. They can give you all the information and necessary information. But for practical training and knowledge, these animals, or you can say this physical lab, should be there, so that students can really learn what is the basic pharmacology. When you are putting an animal onto the any instrument, suppose that you are going with the ectophotometer, you are going with the rotor rod, or you are going with some other experiments of this central analgesic system. There are totally different variations. The pattern of observation is totally different. How the students observe, how they are making the reading, how they're getting the results, how the data is being interpreted, that is totally a different practice. When you are going into the virtual lab, everything is there, you are going step by step. But when you are doing the experiments, there are certain things, means there can be some, you can say, technical points, technical error, calculation should be there, technical calculations are always there. So these things give you the real practical experience. So my opinion is simply that although you can have all the techniques, all the things are there, new softwares are there, virtual labs are there, you can say my cam lab and X pharmacology, they are being used. Means what the students are doing in that case, that they will simply observe what is going on, they will simply see, and they will simply go into the file or the reference book, simply write down. But what they have received, how much information they have received, and how this information will be used practically, means in that case, okay, you have attained knowledge, but where is the application of that knowledge? Application comes with when you are doing the real experiments in the real conditions. You are preparing the drug, because in pharmacology the first important part is preparation of the dose. But in case of software, what we are saying is that okay, I'm taking 3 mg per kg. Okay, that means that you have to select an option, those will be there, and you are going to that intradermal they हो up, and you are going intravenous and all. But when you are doing that, how much drug you have taken, and if there is no any spillage of the drug, real amount of drug is going inside the body, how the onset is there, how much is the duration of action, how is the observation, these are the real, this is the real learning of the students. So in my opinion that software should be there, they can give you the information, but replacing these physical experiments will really hinder the real, you can say, the motive of education, that if you cannot give you cannot give the application to your knowledge, in my opinion that is not considered as knowledge. So for real experiments, the physical lab should be there, and students should be equitized, because in some of the colleges they are having postgraduate courses along with this graduate courses. So means ki then when students are doing postgraduate, students are doing their experiments, at least student can get a glimpse of the animals, they can visit to the animals, they can have some hands-on experience, they can have some practice, that how to give the injections, how to all these things, means these can be demonstrated. Because what is happening these days now, most of the college what they are doing in the postgraduate courses also, is that students are not given to the any animals into the first year of post graduation course, they are given animals into the second year. Means as a result of which, when the students are doing starting their research work now, it will take another 1 to 3 months to get acquainted with the handling of the animal, the methods of blood withdrawal, dose and administration and all. And as a result of which, there is a loss of timing, there is a loss of resources also. So means that if really you want to give some knowledge to the students, it should be a continuous process. It should not be like that okay, then one day you are on the virtual lab, and next day you are saying now you can start with the experiments with the animals. It should be a continuous process, so that student can learn the behavior of the animal, they can really get the information, they can understand why we are doing the experiment, what is the real motto, and if these experiments are performed, how to interpret these experiments, what type of data we can get from these type of experiments. And this real knowledge will come from the practical application, that if you're doing the experiments physical mode, means that you're dealing with the animals, you are going with the preparation of the doses and all these things, data collection, data interpretation, only then you can have some information. Otherwise simply going with the software virtually, you have seen like this nowadays, that we see lots of things are happening, but if you cannot do the experiments your own, it is simply, if I can use that example, virtually we can drive Mercedes, or high speed car at 200 kilometer per hour, 300 kilometer per hour. But is it possible to have 100 kilometer on the road? It is not possible. So virtually you can have everything, but it should be reason, or it should be associated with the physical training also, especially in case of pharmacology. So that is my opinion about that. Uh,
Thank you so much, sir. Definitely, if we indeed agree with you regarding the that we should not only depend on softwares. Practical should be given importance too. It should be continuous mode, and physical lab should be there. And for that, we need skilled persons also. Again, we have come to the point that there should be skills regarding this. So definitely for that, we request all the panelists for in future, if you are planning to doing any kind of skill training regarding this, so definitely do tell us in the Pharma Lok, so that we could also initiate in that program, sir. So we will come to the last panelist of our session, so that at the end we will have a open discussion on at the end. So we have Dr. Rajneesh Srivastav, sir. Sir, we
Thank
have a
you.
last question for you. How do simulation tools prepare students for higher education or research, or they can adequately replicate real-time decision making and procedural accuracy? Whether simply means, sir, whether if a student is learning, for example, any kind of training in software, will it help it in the real-time world?
Thank you, Junaid, for this question, and it's really a very, you can say, challenging scenario for the B Farm students to convince that uh, just hold the animal like this, just input the administration like this. But it is not so much feasible to just, I can say, code the things, because what I have experienced that it is I'm very much agree with the Randhir sir that it is not possible to you can say mimic all the things by just only tell the theory. So what I also you can say experience in the B Farm curriculum, the pharmacy course that will be framed for the pharmacology, that they are just simply you can say enlist that the list of experiments have been you can say mentioned in the B Farm curriculum course, because I'm just focusing my your question because to prepare the students for the higher education, the course curriculum should be aligned like that also. So what I experienced that they are just simply the in the syllabus they are just simply you can say mention the list of experiments that you have to perform, the you can say DRC, or you can say 1 point biopsy, 2 point biopsy methods, and also the route of administrations, and simply write down that these are the you can say the software you have to use, or the experiment have been done by using software also. But what I can say in my opinion that there should be some sort of you can say mimicking models also you have to include that as a in the SIF that क्योंकि क्या होता है कि जैसे कुछ मिमिकिंग मॉडल्स जो हैं जैसे रैट के मिमिकिंग मॉडल्स हैं जिसे हम मैनिक्विंस बोलते हैं, बेसिकली मैनिक्विंस जो होते हैं दे आर यूज्ड बाय द एमबीबीएस स्टूडेंट्स। जो मर्चुरी में जो बॉडीज होती हैं वो तो अलाउड नहीं है, बट एमबीबीएस स्टूडेंट्स जो है दे यूज मैनिक्विंस। So likewise, I also suggest you can say PCI to include the mannequins of the rat, the mice, so that the student they're getting much more closer. You can say anatomical aspect, as well as there are also the mimicking models of rat, mice are also available, so that you can say train the students to you can say how to dose the animals. So I have also some suggestions. Hope so that some of them are agree, some of them disagree also. So the universities or colleges that are having you can say M farm or you can say have animal house, so उसमें हम ऐसा ड्राफ्ट कर सकते हैं कि जैसे अगर कोई एम फार्म का स्टूडेंट है उसको कोई प्रोजेक्ट मिला हुआ है तो why we should not include a B farm student because उसमें फाइनल ईयर में प्रैक्टिस स्कूल और सॉरी प्रोजेक्ट रिपोर्ट होता है। तो अगर कोई स्टूडेंट इंटरेस्टेड है एनिमल स्टडीज में तो क्यों ना हम एम फार्म स्टूडेंट्स के साथ उस बी फार्म स्टूडेंट्स के प्रोजेक्ट को भी हम अलाइन कर सकते हैं, that so that they are having some you can say real time experiences that how you can how the animal house sorry or how the experiments have been you can say proceed. Likewise we have also, you can say, include the BPharm students in a simple, you can say, animal care procedures. जब मैं एम फार्म फार्माकोलॉजी में मैंने एडमिशन लिया था तो मेरे गाइड ने मुझे राइट फ्रॉम द फर्स्ट डे उन्होंने मुझे सारी रिस्पॉन्सिबिलिटीज जो है एनिमल हाउस की दी थी कि यू हैव टू, यू कैन से, टेक केयर ऑल द, यू कैन से, एनिमल्स, उनको फीड कैसे देना है, उनके केजेस कैसे बदलने हैं. सो आई रिकमेंड दैट द, कैन से, द बीसीआई आल्सो, कि अगर आपको मतलब कंप्लीटली बैन तो आप कर रहे हो बी फार्म स्टूडेंट्स को कि आप एनिमल स्टडीज में बिल्कुल आप नहीं कर सकते हैं, बट जिन इंस्टीट्यूट के पास एनिमल हाउस है और वहाँ जहाँ पे बी फार्म कोर्सेज चल रहे हैं, सो अंडर द सुपरविजन ऑफ, यू कैन से, और द परमिशन ऑफ आईईएसी, दैट मे बी शायद कि हम बी फार्म स्टूडेंट्स को बेसिक एनिमल केयर और एनिमल हस्बेंड्री में इन्वॉल्व कर सकते हैं, जिससे कि हम उनको मतलब कि ये बता सके कि एटलीस्ट आप फैमिलियर कैसे, एनिमल्स को हैंडल कैसे करें, रिस्ट्रेन कैसे करें, मींस. तो इससे थोड़ा सा मतलब बच्चों के अंदर इंटरेस्ट भी जगेगा, क्योंकि रणधीर सर ने जैसे बोला कि हम हर चीज को मतलब वर्चुअली हम नहीं समझा सकते बच्चों को. एक्सपेरिमेंट्स हम नहीं परफॉर्म करा रहे हैं, बट स्टिल हम कुछ हद तक हम बच्चों को रियल टाइम एक्सपीरियंस दे सकते हैं. सो और दूसरा मैंने बताया कि जैसे कि जैसे कुछ डिजीज एनिमल्स होते हैं, उनको कैसे रिहेबिलिटेट करना है, मतलब उस जैसे प्रोसीजर्स में हम बी फार्म स्टूडेंट्स को इन्वॉल्व कर सकते हैं. सो दीज आर माई सजेशंस. जहाँ पे भी एनिमल हाउस है वहाँ पे बी फार्म बच्चों को हम इन्वॉल्व कर सकते हैं इन तरह की एक्टिविटीज में, क्योंकि दे आर मतलब एक फाउंडेशनल स्किल्स को डेवलप करने के लिए तो चलिए हम वर्चुअल और ये सॉफ्टवेयर्स का इस्तेमाल कर सकते हैं, बट स्टिल रियल टाइम एक्सपीरियंसेस के लिए वी शुड have to involve the graduate students also. So these are my aspects.
Thank you so much, sir, for giving brief regarding the question that you also highlighted that we should focus on mimicking models and particularly also talk about the mannequins. In MBBS, there are mannequins are used so that they could get more closer to the reality. And similarly, there should be some models in future for the pharmacy, particular to pharmacology also. In that aspect, you also said to one important point in animal rehabilitations. We can include bee farm students in rehabilitation, which is an open house discussion. At the end, one can include many other panelists can include in this. And one more important, you said we can also include UG students plus PG students. Those who are interested, those who are having IEC approval or CCSE approvals, they can include students also. But so now this is an open house debate so that anyone can give us some more include. And so yes, sir, open to all, sir. Any remarks regarding the answer? Everyone is welcome, sir.
Yes, sir. Please unmute your mic first.
Yes,
Yes,
sir,
sir.
you are unmute, sir.
Yeah.
Yes,
Okay.
sir.
Am I audible now?
Yes,
Yes,
sir.
sir,
Thank you.
absolutely, sir.
Yeah, I would like to give a brief comment about the comments made by Professor Satish sir and Dr. Rajneesh ji. Very useful suggestions given by Rajneesh ji that we need to club UG students with PG students, because when we are teaching UG students, not all of them will go towards the pharmacology. So not all of them necessarily may need to handle the animals themselves, and only those who are proceeding towards pharmacology can be, they can be exposed to the actual use of animals when they go to the M-Pharm level. And the UG students, it was a very good suggestion to make them watch when these PG students are doing using animals for their research purpose, which is allowed. So we can make the UG students watch them at least and observe them, so that they can also learn the techniques. And again, one more good suggestion was given by Rajni that use of mannequins. I was on a NAC inspection in Navi Mumbai, and I happened to visit one of the prominent institutions there. They had an excellent setup of these simulated laboratory for medical and pharmacy students, wherein they had used mannequins. Of course, mannequins are very useful way of teaching things when you cannot make use of animals or humans. but sometimes they are very, very exorbitant in their cost, sometimes even cost crores of rupees, which may not be affordable to all the pharmacy or medical college. These pharmacy colleges, of course, they are important and mandatory for medical colleges, but yeah, but we need to think how we can develop those mimic models of animals, where where students could be taught how to handle the animals, if not on the real animals, but at these mimic models. So I would also concur with the Professor Satish sir that as said by Shri Rabindranath Tagore ji, that if you want to cross the stream, you cannot cross the stream by simply watching it or being a mute spectator. You need to jump into it and swim across. Then only you'll be able to cross the stream. That's a well-known quote by Shri Rabindranath Tagore ji. Of course, we need to do that. He was hinting at giving practical knowledge and practical know-how and exposure to students about handling of animals. Yes, but so far as the law itself is, prohibiting us, we cannot break the law. But rightly mentioned by Abdullah sir, that the guiding principle is the 3R principle. And this 3R principle has not been given today or in 2019 when an animal experimentation was banned. This 3R principle has been given way back in 1959 by Russell and Birch. And in which they said, first of all, you try to replace the animal experimentation with non animal methods wherever it is possible to achieve the same. And if it is not possible to replace them, then you reduce their use. Second is reduction, so that which will enable the researchers to obtain comparable levels of information from fewer animals. And if even a reduction is not possible, then you go for the refinement of the methods which will alleviate their pain, suffering, or distress. Because ultimately, the aim is to reduce the pain and suffering to the animals, to reduce the cruelty to the animals, to reduce their distress. And there are laws which prohibit us from doing that, so we cannot overstep the law. So this should be the guiding principle of today's discussion, that we should go back to what was said in 1959 by Russell and Birch. First principle, replacement; second, reduction; and third, refinement. And if we follow that basic principle, I'm sure we will not face any problems or issues. But wherever necessary, animal experimentation has to be allowed for research, and wherever necessary, it has to be allowed for demonstration purpose by the PhD or PG scholars while they are conducting their research to the UG people. Otherwise, simulations have to be advanced, simulations have to be developed, and we need to switch over to the new age. We cannot. We have to keep pace with the fast changing times and trends. We cannot stay behind. Then otherwise, we'll be left behind. So, whole world is moving towards simulated kind of things in research, in training, in manufacturing, in internships. So we cannot stay behind that. We have to keep pace with the changing times and trends, and that way we should know what are the new technologies, new adaptations which we need to. So accordingly, our curriculum has to be refined, revised, revisited, and accordingly, the methodologies and teaching pedagogies should also change. That's my brief comment at this juncture. Thank you.
Thank you so much, sir, for giving brief. So, any other panelists want to add up to this, so that we will go to the conclusion?
As Professor Gir has concluded in a very nice way that all the experiments are necessary and we should also go with the newer techniques. So this is also the need of the hour, that we should be very much acquaintized with the new techniques and all. But along with this, we should also go with the basics of pharmacology. And I think that lots of colleges are there. PG colleges are there. They are running this MPharm colleges also, but MPharm courses. What they are doing is that in some of the colleges, I have that observation that they are simply giving the demonstration to the students. And in pharmacology also, that in the master's degree also, they will simply go with in vitro testing and all. And when these type of students, they are going into the academics or they are going into the industry, because industry, although the pharmacology is very less, but in the cases where they get a job specifically in the department of pharmacology, it will be very difficult for those students to handle the situations, because they don't have any real time experience of this animal handling and all. And along with this that if still they are going into the teacher, that because they have seen the softwares only, they don't know the handling of the software. And if they are going into the academics also after that, means because they have not worked on with the animals and they are not fully uh, trained, or they don't have appropriate technical skills to understand the software and all, again it will affect the quality of education. So means that, as sir has said, that if you want to go with the technology, the first thing should be that there should be a proper training of the technology. Means that you can have a costly car, but if you don't know how to drive it, it is of no use. That is my opinion.
Thank you so much. As I remember, as Professor Satish sir in the beginning told us, we cannot just learn swimming just by watching the videos. So please complete at the end your conclusory remarks, sir. Satish sir is also a mentors in Pharma Lok. Sir, please unmute. Sir, please unmute.
Okay. Am I audible, Junaid ji? Am I audible?
Absolutely,
Yes,
absolutely,
yes,
sir.
yes, sir.
Okay, okay. So I will appreciate the summarization given by Professor Geef and Professor Randhir Sahab. They have beautifully summarized the today's discussion. So in the end, the need of the hour is we should not forget. We should continue our basics. We should continue our traditional approach, whatever is there. But we should also progress. We should also progress. We should also learn the new techniques, especially related to AI simulation. We should also learn because otherwise we will lag behind in the race. So this is the need of the hour. And so the thing is that the pharmacology actually is from labs only. If there is no lab in pharmacology, no practicals are being done in pharmacology. We know that in our pharmaceutical chemistry, pharmaceutics, everywhere practicals are being conducted in the actual labs. So our lab of pharmacology, especially at M-Pharm level, should be animal house. And we should encourage as senior leaders, academicians, we should encourage and promote such types of things that students should participate. No, the need of the hour is also that our education system where the mushrooming of the courses and there is pressure from the management side to admit the students. So there is non-attending trend. So we should not encourage this non-attending trend, as has been rightly mentioned by Professor Randhir Saha, that those students who are not performing any practicals even at M-Pharm pharmacology level, when they come to teach the students, they remain away from the experiment. Similar is the case in other branches also, but in pharmacology, this is very important that we should learn animal handling, dosing, and all these aspects. This is very important. We should discourage the non-attending aspect that is going on. This is damaging our pharmacy profession because new young teachers who are joining the pharmacy profession, they don't have skills in any of the streams. They don't even know how to set the parents in the right position. So this is a serious situation. So we all senior leaders should do something in this regard. Thank you so much. Thank you very much.
Thank you so much, sir. I hope this will reach to the many new range of audience in the pharmacy, and definitely this unethical whatever the process is going on, this should be stopped. And we totally agree with Randeep sir also. And thank you, Gir sir, for including. Thank you, Rajneesh sir, also for giving. Any other for the last one, Tariq sir, Rajneesh sir, any other concluding remarks so that we can come to the conclude part, sir.
I want to conclude. You can say I hope so that this is last. I would just add with the three in the 3 hours that was given by William Russell and also mentioned by Dr. Professor Geer that the replacement, refinement, reduction is there, but we also have to, you can say, take care of rehabilitation, the reuse of animal whenever possible, and the last R is we have to do all these things with responsibility. So these are the another 3 hours that I just want to
Absolutely.
add.
Thank you, sir.
Well said. Well said.
Absolutely, sir.
And from my side, in the concluding remark of Professor Girsar, one very sharp hairline statement is that we can include B.Pharm students to see the demonstration or handling by master's student. Actually, that is the technical point. Actually, because as we at the yes sir also said that because we when we go for the guideline and maybe if we fill, when we fill the CCA form for then there is a point that the persons who are handling animals there should be guideline that they are minimum the PG scholars they can handle the animal so we can include the UG students in the lab to see or to assist the master's student actually that is the point because the technical thing is that the regulatory authority allows only the animal for research purpose not the education so that is the main point so
Right.
we can
Absolutely right.
include that one but the animal handling and all the decision part done by the PG student according to the guideline.
Thank you, sir. I would request Dr. Santosh, sir, to give your remarks so that we could come to the conclusion, sir.
Uh,
Vote of thanks,
Very
sir.
good evening to all panelists. Uh, Discussion of today's topic is very interesting among all the pharmacy uh, fraternity. देखा जाए तो आज का जो हमारा डिस्कशन का पार्ट रहा है ये मल्टी डायमेंशनल हुआ है यहाँ पे एनईपी की बात रही है रिसर्च एथिक्स की बात रही है और दहिया सर ने एक टर्म जो यूज़ किया है क्लासिकल फार्माकोलॉजिस्ट तो वाकई ट्रेडिशनल और मॉडर्न के बीच में जो ये ब्रिजिंग द गैप है जिसके लिए हम बात करना एक लिए बैठे थे कि साइमुलेशन सॉफ्टवेयर के थ्रू हम बच्चों को स्किल्ड करने की बात करेंगे यहीं पे तारिक साहब ने एक बात कही कि साइमुलेशन यूज़ भी करने की बात है तो वहाँ भी ट्रेंड मैन पावर की ज़रूरत है और यहाँ बोलते हुए इस मंच से हमें ज़रा सा भी गुरेज नहीं होता कि एक सॉफ्टवेयर बन गया लेकिन उसको हैंडल करना उसका एसेंस के डेप्थ में जाना इसके लिए वर्कशॉप की ज़रूरत है और मैं इस बात का वेलकम करता हूँ तारिक साहब कि डेफिनेटली फार्मा लोग के मन से एक बार पहले भी हमने एक फार्म को लेते हुए एक डेमोन्स्ट्रेशन कराया था
Yes, yes,
मुझे याद है वो
yes,
धर्मेंद्र
yes,
आहूजा साहब ने खुद ही उसका दिया था लेकिन इसको एक
right,
वर्कशॉप के फॉर्म में हम यहाँ पे प्रोड्यूस करें जिसमें सिर्फ एक ही सॉफ्टवेयर पैकेज की बात नहीं है मल्टीपल सॉफ्टवेयर पैकेज को लेते हुए हम यहाँ पे वो वर्कशॉप मिनिमम 3 days का लेके या इस तरह का ही कुछ लेके चलें ताकि एक स्किल डेवलपमेंट की बात रहे और यहीं पर जो एक थ्रस्ट एरिया जो वाकई है कि सिर्फ साइमुलेशन देख के हम एक्चुअल रियल लाइफ हैंडलिंग नहीं कर सकते तो डेफिनेटली यहाँ पे जो दैया साहब ने कहा मॉडल डेवलपमेंट को लेते हुए या एनिमल हैंडलिंग को लेते हुए ये अपने रजनी श्रीवास्तव साहब ने भी अपना टेस्टिमनी बताया कि एट द एज ऑफ यूजी पीजी प्रोग्राम इनके सुपरवाइजर ने ही एनिमल हाउस की सारी रिस्पॉन्सिबिलिटी नहीं दी डेफिनेटली वो भी एक टाइम रहा होगा जिससे आपका इंटरेस्ट और केयरनेस बढ़ा तो आपने एडिशनल 3 आर जो ये कहा सर रिहेबिलिटेशन रीपर्पज ये रीयूज की जो बात करी तो डेफिनेटली सर ये तो इन द रियल सेंस ऐसा कोई मॉडल हमने बनाया जिसमें कि आफ्टर द वॉशिंग पीरियड एनिमल का हम इस्तेमाल कर सकते हैं तो डेफिनेटली इसको यूज़ किया जाए ऐसा कोई क्रिटिकल मॉडल बनाया जिसमें कि हम उसको रीयूज नहीं कर सकते हैं तो यूथनेसिया वाली बात जो मैंने स्टार्टिंग के प्रिंसिपल्स ऑफ ये फार्माकोलॉजी वाले पार्ट पे एनिमल हाउस को लेते हुए एनिमल यूज को लेते हुए बताया ही था जो कि ऑलरेडी पोस्टुलेटेड है तो यहाँ पे री यूज ऑफ द एनिमल ऐसा कोई एक्सपेरिमेंट हम कर रहे हैं जैसे कि उमन एनएसएड्स के लिए जो एक्सपेरिमेंट्स होते हैं उसमें बहुत ज़्यादा कुछ नहीं है 40 days का वॉशिंग पीरियड रखा जाए तो एनिमल फिर से तरोताज़ा हो जाता है तो उसका इस्तेमाल करें ताकि उसकी लाइफ स्पैन में वो हमारे लिए एक बर्डन और लाइबिलिटी ना बना रहे एनिमल उसका हम इस्तेमाल करें और बेहतर तरीके से दूसरे बच्चे भी दूसरे स्कॉलर्स भी इसका इस्तेमाल करें और इसके लिए रिक्रिएशन वाली बात भी कही जाती है जैसे कॉलोनी फॉर्मेशन में एनिमल को रखा जाए खासकर रैट को जो उसका जो नेचर है नेचर ऑफ लिविंग तो उससे उनका रिफ्रेशमेंट और ज़्यादा इन्हांस होता है ये अपने टाइम में हमने भी करके देखा हुआ था इस पर थीसिस वगैरह भी निकाली थी तो डेफिनेटली सर ये आज का डिस्कशन इन द ऑल एस्पेक्ट मॉडर्न जिसमें कि साइमुलेशन की बात है एआई की बात है और साथ ही जो क्लासिकल फार्माकोलॉजिस्ट की बात है, जिसमें हम एनिमल हैंडलिंग, एनिमल हाउस की केयर की बात को लेके चलेंगे। एक बड़ा वेलकम करने वाला जो एस्पेक्ट आया है, जिसमें कि यूजी के बच्चों को पीजी के स्टूडेंट के साथ एसोसिएट करने की बात, ये बात भी रजनी सर ने ही कहा कि हो सकता है कि कुछ लोग इस पे इत्तेफाक रखें और कुछ को ऐतराज हो, तो ये मैटर ऑफ डिबेट है। लेकिन यहाँ पे डेमोन्स्ट्रेशन बिहाइंड द ग्लास वॉल, हम डायरेक्टली बच्चों को एक्सपोज नहीं करेंगे यूजी वाले को, लेकिन ये जो होता हुआ देखें, इससे ही बहुत फर्क पड़ता है, एक कॉन्फिडेंस लेवल बिल्ड होता है। हम डायरेक्टली एक्सपोज नहीं कर रहे, हम रेगुलेटरी को अवॉइड नहीं कर रहे, क्योंकि उससे इफ एंड बट्स कई सारे आ सकते हैं। तो यहाँ पे पीजी के बच्चे या पीएचडी के रिसर्च स्कॉलर जब एक्सपेरिमेंट कर रहे हो, तो वहाँ पे देखने के लिए हम यूजी के बच्चों को वहाँ पे भेज सकते हैं बैचेज में, जो कि साइलेंटली, जैसे क्योंकि अभी नॉइजिंग को लेते हुए बात रही थी, सिर्फ नॉइज नहीं, वहाँ पे ह्यूमिडिटी और टेम्परेचर को लेते हुए भी बड़ा सेंसिटिविटी रहती है एनिमल हाउस के अंदर एनिमल्स का। तो वहाँ सबका ख्याल रखते हुए, ताकि वहाँ पे बहुत ज़्यादा गैदरिंग ना हो, ताकि इसकी वजह से जो इन्वायरमेंट एनिमल हाउस का जो कंट्रोल्ड इन्वायरमेंट है वो कहीं ना कहीं डिस्टर्ब होता है। तो ऐसे फ्री स्पेस को बिठा के, या फिर हमने सीसीटीवी कैमरे के थ्रू ही रिमोट से भी बच्चों को दिखा के इस बात को कॉन्फिडेंस दिलाना कि आप ही के कैंपस में ये एक्सपेरिमेंट परफॉर्म हो रहा है, तो डेफिनेटली इफ यू आर कमिंग फॉर द एक्सपेरिमेंट, तो आप इसे कर सकते हैं। तो डेफिनेटली सर, आज का डिस्कशन बहुत अच्छा रहा, ये और भी आगे तक जा
And
सकता है।
one more, Santosh ji,
सर,
upgrading the existing software, because lot of flaws and deficiencies are there. So improving and upgrading the existing software, because there is lot of scope for improvement in the existing simulation software.
डेफिनेटली सर, 3D में और ज़्यादा हैंड्स ऑन वाली बात, ताकि एक्चुअल में इन हैंड ट्रेनिंग की तरह बात हो पाए। इसके बारे में भी एक पिछले प्रोग्राम में आपने अपनी ओपिनियन रखी थी, तो ये हमारे दर्शकों को याद होगा, खासकर मुझे तो याद ही है। तो डेफिनेटली यहाँ पे स्कोप ऑफ इम्प्रूवमेंट ऑफ एग्जिस्टिंग
जी
सॉफ्टवेयर, और इसके अलावा भी अब चीज़ें और ज़्यादा एडवांस होती चली जा रही हैं, तो उसके हिसाब से भी रियल टाइम में हम और बेहतर आगे बढ़ें, ताकि बच्चों को समझ में आए। ये अभी अभी जैसे कहते हैं श्रीवास्तव जी ने मिमिक्योस की बात कही, तो डेफिनेटली हम ये सिमुलेशन के थ्रू ही मिमिक्योस को भी लेके चल सकते हैं। कुछ ऐसे एड ऑन्स की बात भी कर सकते हैं सॉफ्टवेयर के साथ, जिसमें कि रियल वर्ल्ड में हम कुछ कर रहे हो, तो वहाँ पे सॉफ्टवेयर के अंदर भी वो चेंज आता हुआ नजर आ रही है। स्कोप काफ़ी ज़्यादा है, और अल्टीमेटली हमें स्किल्ड मैन पावर को डेवलप करना हमारे ये कैरिकुलम का स्कोप है, और उसका नीड भी यही है कि हम एक स्किल्ड मैन पावर को बना पाएं। जब उसे काम करने की जरूरत पड़े, तो वो किसी का शक्ल ना देखे कि कोई और आया हमारी मदद करे। इसमें हमारे सतीश सर ने भी एक बात कही थी, सीआरओज के रोल के बारे में भी जरा सा चर्चा आ गई थी। तो डेफिनेटली कॉन्ट्रैक्ट रिसर्च ऑर्गेनाइजेशन सीसीए भी ये बात कहती है कि आप एनिमल हाउस को कॉन्ट्रेक्चुअल रिसर्च के लिए भी आप कर सकते हैं, लेकिन वहाँ भी डाटा तो आ जाएगा, सैंपल हमने भेज दिया, मॉडल उन्होंने डेवलप किया, डाटा हमारे पास आ गया, लेकिन वहाँ पे स्किल्ड मैन पावर को लेते हुए फिर दिक्कत आ जाती है। तो अल्टीमेटली वो हमारे टीचर बनते हैं, और टीचर यदि इस तरीके से प्रोसेस से आ रहे हैं, तो अपने स्टूडेंट और स्कॉलर को जब रियल वर्ल्ड में ट्रेनिंग देने की बात आएगी, तो फिर वो भी दाएं बाएं की चीज़ें करना शुरू करेंगे। तो डेफिनेटली हम क्वालिटी को बरकरार रखें और उसमें फर्दर इन्हांसमेंट लेके आए, मॉडर्नाइजेशन भी लेके आए, और लेकिन जो क्लासिकल चीज़ें हैं डेफिनेटली वो भी हमारे साथ जुड़ी रहे। और जो फार्माकोलॉजी का या कहिए फार्मेसी का, जब तक कि न्यू ड्रग डेवलपमेंट या न्यू डोजेस फॉर्म का जो इवोल्यूशन है वो रियल लिविंग ऑर्गेनिज्म पे ना हो, तब तक वो सब कुछ वर्चुअल ही है, और रियलिटी जब हमारे बीच में आएगी तो वो और ही रहेगा, हाइपोथेसिस पे काम नहीं चलता। और यहाँ पे एनिमल की बात आएगी तो एनिमल बिहेवियर्स के लिए समझना, जानना और महसूस करना, एक ट्रू रिसर्चर के लिए एक्चुअल एंड पॉइंट तक पहुंचना और उसका जो इंटरप्रिटेशन करना, ये बहुत ही मायने रखता है। ना ही सिर्फ बिहेवियरल एस्पेक्ट, उसके बायोकेमिकल की बात भी करें, हिस्टोपैथोलॉजिकल की भी बात करें, ये सब पूरा का पूरा एनिमल स्टडी अपने आप में बहुत बड़ा रिस्पॉन्सिबल एरिया है, जिसमें कि हम न्यू ड्रग डेवलपमेंट या डोजेस फॉर्म के डेवलपमेंट को स्क्रीन आउट करके हम एक्चुअल ह्यूमन प्रॉब्लम्स को रेक्टिफाई कर सकते हैं, वहाँ उसको अचीव कर सकते हैं। और मॉडर्न और स्मार्ट डोजेस फॉर्म और ड्रग, चाहे वो रिपर्पसिंग की ही बात क्यों ना हो, तो उसमें बड़ा अच्छा रोल प्ले किया जा सकता है। तो इन्हीं बातों के साथ मैं अभी आप सभी पैनलिस्ट का तहे दिल से फार्मा लोक के माध्यम से धन्यवाद देता हूँ। और चीज़ें और भी हैं, तो ये पार्ट आगे वर्कशॉप के माध्यम से आपके बीच में पुनः फार्मा की पाठशाला में हम आगे लेके चलेंगे और उसमें भी आप लोग का इम्पोर्टेंट इनपुट रहेगा, ऐसा मेरा मानना है। तो इन्हीं बातों के साथ आप सभी को पुनः धन्यवाद और साधुवाद हमारे साथ जुड़ने और अपने
Thank you.
वैल्यूबल इन्फॉर्मेशन को
Thank you,
शेयर
Dr.
करने
Sahab.
के लिए।
Thank you for inviting.
Thank you, Dr. Sahab.
Thank you so much
For
to
Professor
all the
Satish
respected
Deepak,
panelists.
Mahmood Badai for taking over
Uh,
as Vice Chancellor.
We have
Satish sir,
already
before I talk,
congratulated sir. Satish sir,
uh,
many congratulations.
Yes, sir.
uh, Thank you and congratulations to Mr.
And
Junaid for conducting very specifically, or you can very excellently this session. Congratulations,
thank you
Mr.
so
Junaid.
much. It means a lot, sir. Thank you, sir, coming from you. And
Congratulations.
I would like to appreciate your time, giving us your time, expertise, dedication. As I told you in the beginning, that I was sure that this is going to be a tradition of excellence that we maintain, and definitely it will help a lot of people back because it will be available on YouTube. So it will give a lot of people a guidance, a thorough recommendations for the future. Also, thank you once again, sir. Thank you so much.
तो
Let's continue to evolve the
सतीश सर
pharmacy.
वापस आ गए हैं।
अच्छा अच्छा
Junaid ji, Satish sir वापस आ गए। So on behalf of Pharma Lok, we
Satish
would
sir,
like to congratulate him for taking over as Vice
congratulations,
Chancellor. Many hearty congratulations.
Vice Chancellor.
Thank you, sir.
Congratulations, sir, once again.
Congratulations, Satish sir.
Congratulations, sir.
Thank you.
Congratulations.
Thank you.
Congratulations to Pharmalok and Dr. Santosh for conducting this program. Not only this program, for maintaining a continuous vibration in our whole pharma fraternity. They both, Amit and Santosh, they are both are inspiration for all of us for their lively, not only lively nature, but live participation and with full enthusiasm.
Of course,
Thank
of
you for the
course.
kind words,
बहुत
sir.
बहुत
Thank
बधाई।
you. With this, sir, thank you so much to everyone, sir. Thank you so much.
thank
Thank
you.
you. Thank you.
Okay, thank you, Santosh sir, thank you, Junaid.